Hello everyone, let's get started. Today we're going to be talking about microbial genetics, DNA replication and mutations. The last time we were together, prior to spring break, we started into this topic. We talked through some terminology, some definitions for important terms like gene and genome and genotype. We reviewed the Central Dogma, which are the 3 statements describing how genes are expressed. We talked about the genetic code and how all organisms use the same genetic code when their cells are building proteins. We talked about how prokaryotic organisms are capable of transferring their DNA both vertically and horizontally. And we discussed the 3 mechanisms of horizontal transmission. We looked at the DNA molecule itself, including the subunit called the deoxyribonucleotide and the sugar, phosphate, and base components of that molecule. We looked at how 2 strands of DNA come together to form a double helix. We talked about this concept of the strands being anti-parallel, with one going in the 5' to 3' direction and one in the 3' to 5' direction. We talked about how the way the bases come together is referred to as complementary binding. And we talked about the hydrogen bonds that hold base pairs together. We also discussed the concept of semi-conservative replication, which describes for us how the DNA molecule is physically copied. And remember, that means that the double helix is first broken into 2 strands, and that each strand is then used as a template for making new DNA. We looked at some images of how that process occurs. We talked about the enzyme that does this important work, called DNA polymerase. And we started talking about the process itself. So I'll begin here today reviewing the first few steps of DNA replication for us. Remember that prokaryotic cells have a circular chromosome. If we took all of the histone-like proteins away from the nucleoid, we would be left with a double helix of DNA in a big circle. Replication has to begin at a point. There's going to be one place in that circle that the process begins, and we call that the point of origin. Now, the cell is going to separate the 2 strands from each other, starting at the point of origin. We refer to the area of separation as the replication bubble. And the replication bubble is going to enlarge in both directions, because we're in a circle, until the entire DNA molecule has been copied. Remember that new synthesis has to begin by priming the old strands. And the cell does that by laying down a short piece of RNA called an RNA primer. The bases in the RNA will be complementary to the bases in the DNA strand, so that piece of RNA will stick to the DNA and give the DNA polymerase enzyme a place to begin making new DNA. After the entire DNA molecule has been copied, the RNA primers will be removed and replaced with DNA so that we'll have only DNA in the new DNA molecules. This is a schematic of this process of replication beginning. Here is the circular chromosome from a bacterial cell. And you can see that we're pointing to one spot where this circle is going to be opened up, where the 2 strands of DNA are going to be separated from each other. The point that we open the circle is called the point of origin, or the origin of replication. And the circle itself, here, is referred to as the replication bubble. Now, as we continue to open the double helix in this direction and in this direction, the replication bubble is going to get larger and larger. It's going to keep opening up in this direction. It's going to keep opening up in this direction until both sides of the bubble meet. And at that point, the entire chromosome will have been copied. Okay. Now, there are some details of replication that can be a little bit confusing, so I want to walk through them together with you. Because DNA polymerase, this enzyme that performs the job of replication, because it can only work in one direction, remember, it can only work from 5' to 3', and when we say that, we mean that it can only add new bases onto the 3' end of the molecule. And of course, when I say bases, I really mean nucleotides, not just bases. So when you open up a replication bubble, because it works in both directions, it opens in both directions, there are going to be regions in that replication bubble that can be made, where the DNA can be made continuously. In other words, the enzyme just adds one nucleotide after another nucleotide after another. And then there are going to be other regions in the bubble where the enzyme can't do that. It has to essentially work backwards, if you will. I'm going to show you an image in a minute that's going to make this a little clearer. But understand that part of each new strand of DNA is going to get made continuously. The enzyme is just going to add one base after another, after another, after another, building the new strand of DNA in a continuous manner. But there will be other parts of each new strand where the DNA is going to have to be made discontinuously. In other words, in fragments. Once the entire chromosome has been copied, the fragments can be joined together so that the new DNA strands are complete. There's no gaps in them. But while it's being made, parts of each new strand have to be made in these fragments. And we give these fragments a name. We call them Okazaki fragments. These were named after the scientist who discovered them. Now, in this image, we can see a double-stranded molecule of DNA that has been opened up for replication. So we have to imagine this right here in the center. This is the point of origin. This is where the double helix is originally opened. And this is the replication bubble. So this bubble is going to keep opening. It's going to open in this direction more, and it's going to open in this direction more. This little region right here, where the bubble meets the part of the DNA that's still in a double helix, that's referred to as the fork, the replication fork. So there's a fork right here, and there's a fork right here. Now, take a look at what's happening inside the replication bubble. At the point of origin, an RNA primer is going to get placed onto the old strand of DNA, the black strand. And the RNA polymerase enzyme is going to come in and start adding bases. It's going to add bases one at a time in this direction. Okay? This is a continuous method of making the new DNA, and we refer to it as leading strand synthesis. If you notice, on the opposite strand of the old DNA, the same thing is going on, but it's going on in the opposite direction, over here, leading strand synthesis. Now, the reason that the enzyme is doing this in two different directions is because the old DNA is anti-parallel. Now, try not to get too swamped down in all these details, but one of these strands of DNA is oriented 5' to 3', the other one is oriented 3' to 5'. So that means because we can only make DNA in one direction, On each of the old DNA strands, the enzyme has to work in different directions. This way on this strand, and this way on this strand. Now that's great. We will get leading strand synthesis going on. But what about these other two areas? This area over here and this area over here. The enzyme can't jump on here and go this way and make a continuous strand while this replication bubble is opening up over here. I know this is a confusing concept, but essentially what has to happen in this region is the enzyme has to start, say, here and start making DNA, and then it has to jump back and start making DNA, and then it has to jump back and start making DNA. As this side of the replication bubble opens more and more and more, in other words, as the fork continues to move in this direction, the enzyme is forced to jump backwards and then move in this direction, making the new DNA. So in this region and in this region, the new DNA gets made in fragments. And this is what we refer to as lagging strand synthesis. It creates Okazaki fragments, and once DNA synthesis is over, those fragments are going to have to be joined together by, you guessed it, another enzyme, and that will create, again, one long continuous strand of new DNA. So let's take a moment to summarize what we've talked about so far. The circular double helix has to be opened in order for DNA replication to begin, and an enzyme will do that for us. An RNA primer has to be placed on each of the old strands. A different enzyme is going to build the primers, and that will begin the replication process. Remember, the DNA polymerase enzyme is going to look for a 3' hydroxy in order to add the first nucleotide. Remember, the 3' means the number 3 carbon on the nucleotide, and there's a little hydroxy group hanging off of there. And this is the place where the enzyme can add the next nucleotide. The enzyme that's doing this work is called DNA polymerase. DNA polymerase is going to build new DNA, but only in one direction. It can only build new DNA in the 5' to 3' direction. So that means part of each new strand will be made continuously. That's what we call leading strand synthesis. And part of each new strand will be made in Okazaki fragments. That's called lagging strand synthesis. Once the DNA double helix is completely copied, the primers will get removed and replaced with DNA by, yes, another enzyme. And the fragments, the Okazaki fragments, will get joined together by yet another enzyme. And that will make the new DNA one long continuous strand. Now, the DNA polymerase enzyme that does this work is pretty amazing. It adds new nucleotides to the old DNA at an amazing rate. And it really does work pretty quickly in that it typically takes a bacterial cell about 20 to 40 minutes to copy its chromosome. If you think about it, that's pretty good, considering there are anywhere from 4 to 8,000 base pairs in a typical bacterial chromosome. Now, it's not perfect because it is an enzyme, it's a biological molecule, and it does make mistakes. In fact, we know that the DNA polymerase enzyme inserts the wrong base about 1 time out of every 100 to 1,000 bases that it places. So once out of every 100 to 1,000 bases, the DNA polymerase enzyme is going to put the wrong base in the new DNA. So instead of matching A with T, maybe it'll match A with G. Instead of matching C with G, maybe it'll put a T there. It makes a mistake. It puts the wrong base in. Now, there is good news in that our cells, all cells, contain what we call DNA repair systems. One of them is referred to as the proofreading system. Once the prokaryotic cell, the microbial cell, or the eukaryotic cell, cells like ours, once they replicate their DNA, the cell will proofread what has just been done. The enzyme literally walks backwards along the new DNA and makes sure that each base pair is correct. If there's a mistake, it gets fixed. Again, most of the time. It's a biological system. It makes errors. So some errors get left in place. Some errors are not fixed. And this is one of the ways that mutations occur. This is a good place for us to stop. We'll pick up this discussion in the next video.